I am Harsha Srivastava, PhD scholar from Jamia Hamdard, New Delhi, India. Also currently working as young professional at the National Institute for Plant Biotechnology, NIPB, New Delhi, India. Here I will be presenting my PhD work entitled Novel Insights into lncRNA mediated waterlogging responsive gene regulation in pigeon pea. Kajanus kajan, commonly known as pigeon pea, is a vital legume crop crucial for the nutrition and economy of billions in developing countries. It offers balanced and nutrient-dense calories and proteins, which is up to 22% compared to cereals, contributing significantly to food security. With a cultivation of about 5 million hectares, pigeon pea is the sixth most significant legume food crop globally. However, environmental stressors, particularly waterlogging, threaten its production, yield, and quality, causing annual loss of 0.28 to 1.1 million tons per hectare and reducing production by 25% to 30%. Waterlogging affects pigeon pea at all growth stages, especially during the seedling and vegetative phases, leading to wilting, senescence, and chlorosis. It inhibits aerobic respiration, hindering growth and development, primarily due to oxygen deficiency and ethylene accumulation, which restrict root growth and lead to cell death. However, the involvement of non-coding RNAs in waterlogging adaptation has not been reported until now. Non-coding RNAs refers to the RNAs that are transcribed but not translated to proteins and play a major role in various biological processes. Long non-coding RNAs, lncRNAs, are a class of non-coding RNAs that contain more than 200 nucleotides and lack protein coding potentials. Previous studies suggested that lncRNAs also play an important role in regulation of various biological processes, including biotic and abiotic stress in different plants. However, to date, there is no systematic report on the involvement of lncRNAs under waterlogging stress in pigeon pea. Therefore, the present study was carried out to identify and characterize the waterlogging stress responsive lncRNA employing a high throughput sequencing approach. We filtered and annotated waterlogging responsive transcripts and then experimentally validated them using reverse transcription quantitative PCR. According to our expectations, qRT-PCR results confirmed that all selected waterlogging responsive lncRNAs respond to waterlogging stress. These results suggested that at least a subset of these newly identified differentially expressed lncRNAs may play an important role in response to waterlogging stress in pigeon pea. These results extend the current knowledge on lncRNAs and important gene expression regulators under stress conditions. The present study is the first report on lncRNA identification and profiling under waterlogging stress in tolerant and susceptible genotype of pigeon pea. So, in the analysis pipeline, RNA sequencing of leaf sample of two waterlogging tolerant, which is JVP110B, and susceptible genotype ICP7035 under control was done by Illumina HiSeq 2000 platform for building a reference transcriptome. RNA-seq produced 479 million raw reads as paired-end. FastQC was used for quality control of raw reads. Trimomatic used for adapter sequences and low quality tags, resulting in 467 million clean paired-end reads for transcriptome assembly, which were done using Trinity with default parameters, resulting in total of 47,665 non-redundant assembled transcripts in pigeon pea. All transcripts longer than 200 base pairs were retained. Venn diagram of putative lncRNA screen via coding potential analysis method include BLASTX, CPC2, Interpro, Transdecoder, and RFEM database. Number within circle represent the number of lncRNA transcripts predicted to be positive. The intersection of the 5 circles is taken as prediction result. Finally, we identify a total of 12,669 long non-coding RNAs. To identify differentially expressed lncRNAs under waterlogging treatment compared to control, we analyzed using HR package, identifying transcript with log to fold change and FDR less than 0.01 as differentially expressed lncRNAs. Here, the Venn diagram in the identification putative lncRNA section reveals 645 and 443 significantly differentially expressed lncRNA between tolerant and susceptible waterlogging genotype respectively. In the differential expression section, volcano plot displayed a different number of differentially expressed lncRNAs and mRNAs in tolerant and susceptible genotype, with blue circle denoting significantly upregulated and orange circle denoting significantly downregulated transcript with p-value less than 0.05. Box plot visualize the expression level of lncRNAs and protein coding RNAs in tolerant and susceptible genotypes. To understand the function of differentially expressed mRNAs, gene ontology and Keck pathway enrichment analysis were also performed. So here in section of clustering analysis, for functional studies, we identified and selected lncRNAs of Pigeon P's waterlogging tolerant genotype. We used lncTAR tool to predict putative binding events between differentially expressed lncRNAs and their trans targets to sequence complementarity. We observed diverse gene ontology terms enriched by target genes in each comparison, indicating difference in response to waterlogging stress between Pigeon P genotypes. In the tolerant genotype, target genes were enriched in processes resulted in related to oxidative stress response, amino acid export regulation, intracellular protein transport, and oxygen signaling. These processes enhance stress perception, cellular redox homeostasis, and oxidative stress tolerance. Key genes identified during ribulose include ribulose bisphosphate carboxylase oxidase activase, heat shock proteins, glutathione S transferase, and peroxidase, which contribute to oxidative stress tolerance. Notably, transcription factor families such as Virki, ARF, and MIB were also significantly enriched. Heat map showed expression pattern of selected waterlogging responsive lncRNA and mRNAs compared to control under waterlogging treatment. In plants, miRNAs, microRNAs target both lncRNAs and mRNAs, reducing their expression through RNA cleavage. lncRNA can also act as sponges, sequestering microRNAs and indirectly modulating mRNA expression. To understand this competitive endogenous RNA, which is cERNA mechanism, we constructed a cERNA network integrating microRNAs and differentially expressed target from the Pigeon P waterlogging tolerant genotype, generating interaction network using cytoscape. These interactions revealed the complexity of regulatory pathways under waterlogging stress in Pigeon P, highlighting the need for a comprehensive understanding of these processes to evaluate genotype tolerant to waterlogging. To validate our finding, we selected 12 ETMs, endogenous target mimic, and their target for QRT-PCR. Our study sheds light on the gene expression regulatory mechanism during waterlogging stress in Pigeon P, providing valuable insights into its molecular response to these environmental challenges. Thank you.